I'm Rizwan, third year MBA student in high tech IMS Texla Kent. So today we are going to start with the topic of neuroanatomy. In neuroanatomy, first we have to cover the basic concept of nervous system, in which we will divide the central nervous system into two systems, the main function. So before we are going in detail of the nervous system or the neuroanatomy, we have to divide it in a parts. So me that we have the basic concept of each part here. The first thing we divide the nervous system is in two types. One is central nervous system. The other one is peripheral nervous system. The central nervous system includes brain and spinal cord, and the peripheral nervous system further include somatic system and the autonomic system. If we are firstly discussing the central nervous system, in central nervous system we have brain and spinal cord. What we have in brain and in the spinal cord, right? So in the brain and spinal cord, there are two major structure which we are going to discuss, and I must say you have the peak level concept of these two things, right? What are these two structures? In the central nervous system, and I mean in the brain and the spinal cord, we have two things which we are going to gonna maybe you have basic concept of them. One of them is neuron, and the other thing is of neuroglial cells. Now, what is neurons and what is the neuroglial cells? Neuron is that component of nervous system which conduct action potential, right? And what is the neuroglial cells? Neuroglial cells is the supporting cells. These are the cells which make up the structure of neurons, right? And these are bundle of connective tissue. So we have two basic structure. One is neurons and the other is neuroglial cells. In central nervous system, or we can say in the brain and spinal cord, the main two components we have to discuss, or we have the basic knowledge of it, is neuron and neuroglial cells, right? The function of neuron is to conduct action potential, to conduct synapses. And what is the function of neuroglial cells? These are the supporting cells. After that, we divide, we see what is present in the neurons, or what is neuron. As we already said, neuron is for the conduction of action potential. So now we have to divide the neurons. What contain neurons? In neurons, we have two structure. One we called it gray matter. The other one is white matter, right? Neuron, when we have to divide the neuron, we say it has gray matter and it has white matter. So what is gray matter and what is white matter? The collection of cell body of neuron is called gray matter. I have write it in very simple words, right? What is gray matter? Collection of cell bodies of neuron, we call it gray matter. And what is white matter? The axons of neuron is called white matter. So you have to remember then what is gray matter. Cell bodies of the neurons is called gray matter, and the axons of the neurons is called white matter. Then we have to further to divide gray matter and white matter. As I'm teaching for first time, right? Now we have cell bodies. Cell bodies of neuron is called gray matter. So in this division of gray matter, we have two types of gray matter in whole nervous system. One gray matter is called cortex, and the other one is called nucleolus. Now, why we called some gray matter are cell bodies of neuron cortex, and why we other called nucleolus? It is differentiated on two bases. The cell bodies of neuron in the central nervous system, which is present on the surface of central nervous system, right? It is present in cerebellum surface, it is present on cerebral surface. The cell bodies of neuron which are present on the surface of the central nervous system are called cortex, right? The other neurons cell bodies which are present deep in the central nervous system, right? And surrounding the white matter are called nucleolus, right? So we have two types of cell bodies, two types of gray matter. One, which one we called cortex, the other one we called it nucleolus. We can see here. It's a bit difficult for me. Right, you can see here. These are on the cerebellum. Cerebellum surface, we called it cerebellar cortex, right? These are the gray matter. And here in the cerebral cortex or the cerebral hemisphere, you can see on the surface we have cerebral cortex, right? What is it is? It is the gray matter. And you can also see within the central nervous system or within the deep structure, we called it nucleolus. And the gray matter which is present within the forebrain we also called as basal nuclei, we called it nucleolus, right? So we have gray matter, we have two types of gray matter, one present on the surface of the cortex or surface of the central nervous system, the other one which is present deep in the central nervous system. The gray matter which is present on the surface of the cerebral cortex, we called it cerebral cortex, cerebellar cortex, depends on the location. The other which is present deep in the central nervous system, we called it nucleolus. So this is the division of gray matter. We have two types of gray matter. Then we have white matter. So white matter also classified into three types. What is exactly white matter? We have firstly call it collection of axons of the neuron is called white matter. In a very simple words, what is white matter? Collection of axon is white matter within central nervous system. What is the function of axons? They also conduct the information, conduct the action potential from one neuron to other neuron. So we have three types of axon. The one we called it tracks, the other one we called associative fibers, and the other one we called is commissure fiber, right? As axon, the function of axon is to conduct the information, the action potential from one part of the brain to other parts of that type of axon, that type of white matter which conduct the information from upper side of the brain to lower side of the brain, or we can say from brain to spinal cord or from spinal cord to brain. That type of axon is called track. And we see later on tracks are of two type, ascending tracks and descending tracks. Achha. The other type that is associative fiber, that type of axons which conduct the information from front side of brain to back side of brain. ठीक है ना? Occipito frontal. So this is called the associative fibers that connect anterior posterior side of the CNS back to front. The other type that is the commissure fiber. These commissure fibers are also the type of axon or white matter which connect left side to right side, right? As the example, we have corpus callosum, which connected the right and left hemisphere. So in white matter, we have three types of white matter within the central nervous system. One we called it tracks, which connect the upper and lower portion of central nervous system. The other one is associative fiber, which connect the anteroposterior side of the CNS back to front. The third one is commissure fiber, which connected the left side of the CNS to right side of CNS. As example is corpus callosum. So we have three types of axons or white matter. We have two types of gray matter or cell bodies. Then the other thing is what is neuron and tracks? It's very important to know what is neurons, not neurons. What is nerves and what are tracks? What is the difference between nerves and tracks? So the basic difference between nerves and tracks is that nerves is also collection of axons and tracks are also collection of axon. But why we call it nerves and tracks? So the collection of axons in the peripheral nervous system, we called it nerves. And the collection of axons within the central nervous system, we called it tracks, right? So the collection of axon has two names. In peripheral nervous system, it has different name. In central nervous system, it has different name. In the peripheral nervous system, we called collection of axon as a nerves, and in the central nervous system, we called the collection of axon as a tracks. As already we learned that tracks is the type of white matter. Then the other thing, what is the myelination of central nervous system and the peripheral nervous system? So in the myelination, we have different cells which myelinate the peripheral nervous system and the central nervous system. So in the peripheral nervous system, we have two cells which myelinate the neurons. One of them is called Schwann cell, and the other cell is the satellite cells. While in the central nervous system, we have four cells that myelinate the neurons. Why we require myelination in neurons? For the faster conduction of the action potential or the information. The first cell is astrocyte, the other one is oligodendrocyte, and the third one is microglial cells, and the fourth one is epidermal cell. So these four cells are used to myelinate neurons in the central nervous system, and these two cells, Schwann cell and satellite cells, are used to myelination to cause myelination in the peripheral nervous system. It is also MCQs. It is also asking the question which are the cells which are used to myelinate peripheral nervous system and what are the cells which are used to myelinate the central nervous system. Here you can see in the central nervous system we have four types of cells. The one is astrocytes, the other one is oligodendrocyte, then we have microglial cells, and the other one is ependymal cells. So these are the four cells which are used to myelinate the neurons in the central nervous system. And then also in the peripheral nervous system, we have two type of cells. That one is Schwann cells, and the other one is the satellite cells. These cells are used to myelinate the neurons in the peripheral nervous system. So why we need myelination? The only and the basic answer to that question is we need myelination to conduct the information faster. So in today's lecture, I just put these information are just these 2 or 3 points for your convenience. I'm going to revise that point. First, we have discussed central nervous system and then peripheral nervous system. In central nervous system, in our first day or the initial day, as it's my first time I'm speaking English to you people, and during my teaching as I'm Urdu speaker, as I live in Pakistan, so it's also a bit difficult for me to punctuate to you people. So in that lecture, in today lecture, we just covered very basic few 2 or 3 things. Inshallah, today we will cover a lot, and our lecture will be of 40 minutes. So I'm revising that again. Central nervous system, you have just two structure. You have to remember two structure. Number one, neurons, and number two, we have neuroglial cell. What is the function of neuron? To conduct action potential. And what is the function of neuroglial cells? It is the supporting cell. It is a connective tissue. Then in the neurons, you have divide neurons into two types. We have gray matter and we have white matter. What is gray matter? In a very simple word, in a two words, you have to remember that is cell bodies of neuron is called gray matter. And what is white matter? The axons of a neurons is called white matter. You know, neurons have two parts. One is called dendrite, which conduct the information towards the neuron, and one is called axons, which conduct the information away from neuronal cell bodies from one neuron to other neuron. So the main two things, the very basic concept of neuroanatomy is what is gray matter, what is white matter. If you know the exactly crystal clear concept of gray matter and white matter, it will be bit easy for you to cover the other aspect of the neuroanatomy. So what is gray matter? Cell bodies of the neuron. And what is white matter? Axons of the neuron. As we have divided the gray matter or cell bodies of neuron further into two types. One is cortex, that cell bodies or gray matter of neuron which is present on the surface of central nervous system. It is present either in forebrain, in cerebellum, or in other parts of the forebrain. And the other one we called is nucleoli or nucleolus. that part of cell bodies or that cell bodies are pieces of gray matter which is present deeply in the central nervous system and surrounded the white matter. We called it nucleus. Then after the gray matter, we have white matter. You can see here the different gray matter types. Then we have white matter. Also, we divided the white matter into three types. That first one is the tracks, which connect the upper and lower part of the brain or the central nervous system. Then the other type is associative fibers, which connect the anteroposterior side of the central nervous system, or we can say back to front of the brain or the central nervous system. And the third type we called it commissure fibers, which connect the right and left side of the nervous system, or we can say brain or the spinal cord. Right? That function is we called it the white matter. And the example is corpus callosum, as corpus callosum is the present in midway between the two cerebral hemispheres, and it is the connector which connect the right and left hemisphere and transmit information from one part to other part. After that, we have see what is the difference between nerves and tracks. It is also very common question to be asked by any neuroanatomy student or anywhere in the web, or it is a very tricky question. What is the difference between nerves and the tracks? So nerve and tracks is the same but in different system. Nerves is also collection of axons, and the tracks is also collection of axon, but nerve we called collection of axon in peripheral nervous system as a nerve, and we called connection of axon in central nervous system as the tracks. And we have further three types of the tracks. Then myelination of the neurons. What is the cells which are used to myelinate neurons in peripheral nervous system? These are Schwann cells and the satellite cell, and there are four cells which are used to myelinate the neurons in the central nervous system. So for today, these are the very basic and very short lecture, as I'm making practice how to deliver lecture to international students as it's my first time. I hope you will understand a little bit, maybe 0.1%, but I tried my best to deliver that lecture. Inshallah, in upcoming days, it will be more convenient for me to deliver the lecture, and I have to put in slides more content, so we may have 40 minutes lecture. Thank you. Any question till now? You can stop the lecture recording.